Microorganisms live all around us and many in us as well. Well, how does this work? There are three types of symbiosis, or living with something, called commensalism, mutualism, and parasitism. Commensalism is when one benefits and one is neutral. Mutualism is, as the name sounds, it is mutual, so both parties benefit. An example is Vibrio fischeri and the Hawaiian bobtail squid, which I'll talk more about later, but here's a nice drawing. And parasitism is where one is damaged, the host is damaged, and the invading microbe is parasitic to it. It benefits from hurting the host. But how can they benefit from living in harmony? A great example is our stomach, which has very specific microbes that are there to help digest foods we otherwise couldn't digest without their help. And by having them there, we can eat a wider, more diverse diet. Speaking of food, bacteria that are nitrogen fixing are very important agriculturally. They are in the soil and take in nitrogen gas from the atmosphere. And through a process called nitrogen fixation, they turn it into an NH3 ammonia compound that is able to be uptaken by the plants. A bacterial example of this is the Anabaena, which forms under nitrogen starvation, as in when the ground doesn't have nitrogen in it, it forms heterocysts amongst its cell chain here, which are 10 spaces apart, give or take. And they're a specialized cell that takes in sucrose, food source. This takes a lot of energy to do. Nitrogen gas from the atmosphere, and NADH, a component in the energy making process. And the output is glutamine, or 2NH3, which is what gets put back into the soil, which is a form of nitrogen in the soil that the plants can use. How do they talk to each other, though? Can cells even do that? Or can bacteria talk to each other? Yes, they can, through something called quorum sensing. The first diagram here shows intraspecial counting, we'll call it, where each cell of the same species creates a specific compound it excretes into the environment. Neighboring cells can sense how much of that secretion is around and then determine how many quote-unquote siblings are around. There's also a different secretion that is kind of like an overall head count, where every single bacterium basically can universally make this one compound, and it's a way to take a head count of the entire bacterial environment. A great example of this is in Vibrio fischeri, which is what I mentioned when talking about the mutualism type of symbiosis. This bacteria can glow in the dark. It lives inside a specialized organ of this deep, this squid that lives on the shores of Hawaii. Basically, when enough of these cells are in a contained environment, they, through quorum sensing, activate a group choice, a group activity, which is in this case to bioluminate. If there are not enough cells in a certain container, they don't activate. Well, how do bacteria get into us, or any living being, really? Portals of entry, areas that lead from the outside directly into the body, like the nose, cardiovascular system, exposed organs, cuts, even the ears, any area that can get into the body is potential to be infected. The skin is least likely to, it's our protective barrier, but if it's damaged, then it allows for bacteria to get in. But how many is bad? There are two mathematical values to analyze to determine this. It's ID40, which is ID50, sorry, which is the amount of pathogens needed to express infection in 50% of an exposed population. LD50 is similar, but it's about lethality, not just infection. Same premise, though. However many it takes to express lethality in 50% of an exposed population. Once they're in us, how do they do anything? Well, bacteria have adhesins or ligands on the outside of their cell membrane that interact with host cell wall receptors that can give them special access or use of host cells. They can form biofilms or capsules and slime layers to make it difficult to wash them away via physical mechanisms, and can make them resistant to antibiotics and disinfectants. It can make them hard to phagocytize when cells will engulf invaders to basically kill them. They can stick with special proteins that can adhere to this host cell wall, or they can just grab on with fibrae or pili, shown in the drawings down here. The last two things we're going to talk about are related to how the body fights invaders. Just like how the flu, every year they need to make a new vaccine. That is because of antigenic variation. There are antigens on the outside of the pathogenic cells that are recognized by the immune system every time it comes back. If there is antigenic variation, there is some kind of rapid mutation that happens on a certain time frame that creates new antigens, thus confusing the host's immune system and letting the pathogen pass by. There's also a specific type of toxin type called AB toxin. This is a mechanism by which toxins from pathogens get into cells. It can either adhere to the surface, and part B acts as the carrier. Part A is the actual toxin, this orange bit. Part B can stick to the surface and just let part A in, or they can be engulfed in a membrane by pushing inward. Once they're inside, then part B lets out part A. Thank you.